From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 16th, 2023. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin of the University of Puget Sound in Tacoma, Washington. Today is the 12th episode of Matters Microbial. Thanks as always for watching and or listening. First, as always, I like to show you a member of my Wunderkammer, my cabinet of curiosities. I've long been fascinated by meteorites, the idea of holding a piece of what seems like rock or metal that's been drifting through space for millions of years really gives me pause when I'm feeling low. Perspective. This is a specimen, let me see if I can get the name right here for you, of the Sikate-Alin fall that took place in northern Russia in the late 1940s. I put a link in the show notes to this wonderful meteorite specimen. It's really amazing stuff. Today's inspirational lapel pins. Everyone knows I love me some Halloween, and I like to tell people that I wish it was Halloween all year long. Hence the pin. Also, in keeping with the idea of meteorites, the idea that every atom in and of us was forged in the heart of a dying star is simply amazing. Hence the second pin. By popular demand, I wanted to introduce the mascots of my courses and my little laboratory where my students and I work. Baby Carl is a 4-year-old Kenyan sand boa and is sweet beyond words, really a teeny tiny snake. Lady Azula of the Fire Skink Nation, on the other hand, is full of attitude. She's a 2-year-old African fire skink. They're both such interesting creatures, and my students really seem to like meeting them. Kind of emotional support reptiles. Also, we discussed ice nucleation bacteria a few episodes ago. Remember? There's in fact some evidence that bacteria can nucleate snowflakes as the weather gets colder. Think about that. So a scientist artist named Red Black Pen created this fun cartoon of the process showing bacteria riding snowflakes down from the sky. This in turn led my friend Dr. Anestes to 3D print me some fluorescent bacteria riding snowflakes. As always, microbiology for me is always filled with OMG and WTM. I had an interesting question from a listener or viewer that I wanted to share. It was sent to me in the Matters Microbial account. How many bacteria are in a bacterial colony? So I had a new Doc Martian research student, Ariana, explore that question for you. First, here is a colony before we did anything. How many cells are in that colony? Ariana used a biopsy punch to punch out the entire colony and its surrounding agar surface. Resuspended it. As you can see, she did a dilution series, and then went ahead and plated out samples from that dilution series. Now, one advantage to doing this with bioluminescent bacteria, as we did, is that it's very clear to see what's taking place. After all, look at, for example, how a typical plate looks, with and without. So in other words, you see it with the light that's incident in the room and also by its own light. It's going to be a topic we're going to discuss today with our guest as well. And then in addition, here's a dilution series. And this is something that really captures the student imagination and actually makes it easier to learn basic techniques in microbiology. So Ariana was able to determine there were about 1.26 times 10 to the 8th bacteria in that tiny colony, which is simply amazing. And as you will see later today, my research students often reflect my somewhat unusual demeanor, as you can see when the plating was taking place using a Bunsen burner and Ariana. The 8th week of my microbiology class is moving forward. Single colony PCR reactions from our marine microbiological safari samples have been carried out, checked successfully on DNA gels, and cleaned up and prepared for DNA sequencing. It's a very exciting time. We also continued to look for possible antibiotic-producing colonies, and we had a little bit of trouble with that in the last week. So I had to redo some work. And if you look carefully here, I've indicated possible antibiotic-producing colonies you find from lichen, possible antibiotic-producing colonies from moss, and possible antibiotic-producing microbes from soil. And it's a good thing I used fungicide this time. Finally, looking at wild microbes from the environment can be challenging. Have a look at this swarming microbe from the soil. It takes over plates in a flash. One of the things that makes me feel really good about my career has been the number of my students from the laboratory who've gone on to earn PhDs. I'm currently at 27 such Doc Martians, as they're called, 5 of whom hold faculty positions. There is literally nothing more rewarding to me than having such students stay in touch and, in some cases, help me with research. I create my own colleagues. And what I really am, to be fair, is a microbial talent scout. These are wonderful people, and I'm privileged to be part of their journey. Some years ago, I had a student in my Biology 111 introductory class named Ruth. I wrote this note on one of her exams. After a couple of years, Ruth indeed joined my lab, as you can see here, next to some microscopes. Now, as I have mentioned, my personal style tends to select for students that are a little bit similar to me in our viewpoints, and Ruth is no exception, as you can see when she's wearing this mask. I also will never forget taking Lux photos of students. Here's a nice one of Ruth and another student from her time here at the University of Puget Sound. But this Lux photo of Ruth that I genuinely took and have not altered really made me worry that Ruth was a demon. When I asked about it, she said nothing. Regardless, I'm proud to have had her in my classes and in my laboratory. After graduating from the University of Puget Sound, Ruth was a technician for Dr. Kat Milligan-McClellan at the University of Alaska, at that time studying the microbiota of stickleback fishes. Following that, Ruth decided to work on her PhD with the quite wonderful Dr. Mark Mandel at the University of Wisconsin-Madison, studying the fascinating symbiosis between the Hawaiian bobtail squid, Euprymnus scalopes, and its bioluminescent bacterial partner, Vibrio fischeri. After earning her PhD this past summer, Ruth has just joined the lab of Dr. Juliet Willett in the medical school at the University of Minnesota to do postdoctoral work on biofilms and gram-positive bacteria, and it is such a pleasure to introduce Ruth to the Quality Quorum for today. Hi, Ruth.
Hello, it's so nice to be here.
I'm so glad you're here. How is the weather over there today?
You know, it's a little chilly. Haven't gotten any snow yet, so I'm thankful for that. It's feeling like fall, and I'm okay with that.
Oh, yeah. Oh, yeah. Well, we have a lot of things to talk about. I kind of promoted you as a first-generation student because you, like me, are one. Do you have anything you'd like to say about what made you become a scientist?
You mostly. So yeah, I'm first generation. My parents didn't go to college. I've always been, I guess, of the scientific mindset. Like growing up, I was always, messing around with things, trying to figure out how things worked. And my parents are like, "Oh yeah, science. You should go into medicine." I was like, "Yeah, cool. That sounds great." And then I got to University of Puget Sound, which is a small liberal arts college, primarily undergraduate. It was the exact environment that I needed to survive college, truthfully. I see these large universities that I've been to for grad school and such, and I'm just like, I couldn't do it back then. So small liberal arts colleges are where it's at for me. And that just really fostered the kind of environment where I could, explore science and get to know scientists such as yourself and see that doing scientific research isn't a scary thing. I distinctly remember my first year being like, oh, people are doing research? I have zero idea how they could possibly be coming up with completely novel ideas all by themselves. And it turns out it's not like that exactly, especially if you're an undergraduate student. So, talking to you, Doc, and other scientists and my other peers that were actually doing research really got me to understand what research was like.
It truly is a mindset. And one of the things that I like about teaching first year students is I can usually identify people that have, I don't know if you want to call it the quirk or the bump, whatever it is you want to call it, that means they want to know what happens next. And you were certainly like that. It wasn't enough just to say, okay, here's a fact I have to know. And actually, no student talks that way.
Mm-hmm.
But some students, they just have so much on their plates, which is always funny for microbiologists to say.
Yeah.
But in any event, you always wanted to know what the next thing meant. And when I knew you, you were pretty well convinced that you wanted to be a medical type or health professions person. And I bided my time because soon I knew that your love of science would take that over. And in fact, it did. And it made me very happy.
Yes, it did, and you were correct. When you wrote that on my first exam in the intro class, I was like, psh, no way, I couldn't possibly. And then senior year, I took your microbiology class, because at that time you were already my academic advisor, and
yeah,
I enjoyed learning from you. So I was like, okay, I'll take the micro class, and that really did it for me.
Well, and also you found an environment where you could be quirky.
Mm-hmm.
I mean, every laboratory has a different environment, and one of the tricks is to find a place where you fit in.
This is true. I remember the micro labs were the only lab that I came back to, like, at not lab time during the week, just so I could, like, see the plates and see what happened.
Well, that's exactly what I meant. You want to see what happens next.
Mm-hmm.
And that's the sign right there of that terrible addiction to learning. But
Yeah.
that here you are. So if it's okay with you, I'd like to kind of start this off. I mean, one of the things that's been made clear to me is that when there's a subject that someone loves, they sometimes get tangled up in jargon. And it's okay between scientists, but I believe very strongly we have to present what it is that we're talking about to people who perhaps don't have our background. I don't mean people of like X or Y or Z. I just mean people that aren't as interested as you are. And considering most of science is supported by the government, I think it's really important for us to be able to do that. As you recall, when you took microbiology from me, I would ask students to create a one-page version of their term paper for non-STEM majors. And I thought that was a very important thing to be able to do. So you went on to graduate school at one of my favorite places, the University of Wisconsin-Madison, and you decided to work in one of my favorite systems involving symbiosis, which is between the Hawaiian bobtail squid and Vibrio fischeri, although they're trying to change the genus name. So I'm just going to call it Vibrio fischeri because I'm old.
It's
Is that okay with you?
None of us call it the other name, so it's fine.
Yeah. But in any event, the advantage to this system, and I put a lot of stuff in the show notes. I've put the Ed Yong video about the squid-Vibrio symbiosis up there. I put a couple of review papers. I put one of your papers up. Oh. But I think it would be good if you talked a little bit about that symbiosis for people. Would you mind?
Yeah. So the symbiosis between the Hawaiian bobtail squid and Vibrio fischeri is actually what got me like super interested in microbiology, because it's one of the first things you talk about in micro class. Because as you know, it's just like a super awesome system. And if that doesn't sell you on microbiology, I don't know what will. So the way this symbiosis works kind of in general is that there's this tiny squid about the size of a golf ball. Can throw up a photo here of a squid. that lives off the coast of Hawaii. And we're able to find them in like pretty shallow water, actually. So they're nocturnal when they come out at night to hunt for food, like shrimp or little fish or something like that. There's moonlight that shines down on the squid, and the shadow that's cast by the squid and the moonlight makes them rather obvious to predators. And what Vibrio fischeri does is that it helps hide the squid. And it does this because Vibrio fischeri is a naturally bioluminescent bacteria that lives in the ocean. And by bioluminescent, I mean it glows in the dark, which is just the coolest thing. The thumbnail is where you can see Vibrio fischeri glowing on a plate, which is never not cool to look at. So there's the specialized organ inside the squid called the light organ, appropriately named, where Vibrio fischeri colonizes, and it's the only bacteria that lives in that organ. So it's highly specialized organ where there's basically the one bacteria, one host interaction. We really like this binary system in terms of studying host microbe interactions because it makes it a lot simpler for us to dissect factors that are required for the association. Vibrio fischeri colonizes this light organ, and that's where it produces the bioluminescence that helps camouflage the squid. There's a couple ways that we believe that Vibrio fischeri is helping camouflage the squid. So one is that if there are predators looking down from above at the squid or at the water in general, then the glowing from Vibrio fischeri from like under the squid, if the glow is going down, it hides the shadow so that the predators can't see the shadow as obviously. And then the other version I've heard is that if there's anything from below the squid, that the light shining down on the squid looks like the moon, so that anything from below can't see the squid as well either.
So it's interesting to me. I mean, this is something that I've known about for many years because of my friendship with a lot of the people that work in squid-Vibrio symbiosis. But if we were to study any type of symbiosis, if it's always together, say the way that our mitochondria are with us, it's very difficult to study that because you can't separate both members effectively.
Mm-hmm.
With the squid-Vibrio situation, you can grow Euprimna without the bacteria. You can grow the bacteria without the squid. And so that makes it very easy to work on. And a lot of very, very wonderful and honestly quite sharing people have created this whole system where they can begin to manipulate both sides of the equation to determine what's important. So can you tell me how it is this bacterium is able to establish itself? Oh, before I say anything else, what happens to the squid if it doesn't meet Vibrio fischeri?
Oh, that's a great question. That's actually the question that I asked Mark when he was giving a presentation for us first-year PhD students. And long-term, I'm not really sure what happens. I know one of the things that Vibrio fischeri does in the squid is that it stimulates this developmental morphogenesis. So when the squid newly hatched squid, they have these appendages just outside the light organ, just like these appendage things. We call them ciliated appendages. And those appendages basically help Vibrio fischeri get to the light organ. And then once Vibrio fischeri is in the light organ, those ciliated appendages go away. So that's one of the things that happens with the interaction. So without the interaction, you can imagine those ciliated appendages would not go away. But in terms of long-term effects, I'm actually not that sure. And that is also the answer Mark gave me when I was first year.
But it is interesting to note that you have this whole developmental program. This light organ is not simply a pouch. It's got structure to it.
Mm-hmm.
And it's responding to signals from the bacterium switching on that particular program, which makes this of interest not just to bacteriologists or microbiologists, but also to people interested in animal development.
Yeah, definitely. And it's very much a dialogue between the bacteria and the host, where the host is sending the bacteria signals so the bacteria can do its thing, the bacteria is sending the host signals so the host can do its thing. So there's definitely communication there that we're still working on uncovering.
You know, Ruth, it's funny you would put it that way with the dialogue, because in graduate school, back when dinosaurs ruled the earth, I worked on the symbiosis between Rhizobium melilodi, a nitrogen-fixing bacterium, and alfalfa. And indeed, both sides of this, they form root nodules that can fix atmospheric nitrogen into ammonia. It's the basis for crop rotation. But what was interesting here is a lot of similarities to what you're discussing with the squid-Vibrio symbiosis. There's a dialogue between both members. There's a specialized structure that gets formed in reaction to meeting the bacterial partner. And there are changes that take place within. But once again, both partners talking to one another using chemical signals.
Yes.
Do we know anything well, there's so many questions. Do you mind if I ask this, or would you rather just talk about your own experiences with it?
Ask away.
So could you tell me what it is that the squid is doing to keep other bacteria from invading the light organ?
Yeah, so at least during the establishment of the symbiosis, there's several different steps that happen that help ensure that Vibrio fischeri is the only thing that's getting in. The first general step that we think about is biofilm formation by Vibrio fischeri. So just outside the entrance to the light organ on those ciliated appendages that I mentioned earlier, Vibrio fischeri produces this specific type of biofilm called symbiosis polysaccharide, or SIP. And it produces that biofilm mostly at that stage, and it helps the Vibrio fischeri stick to each other, and it's also sticking to the ciliated appendages, and that biofilm kind of excludes other bacteria. And then from there, the bacteria swim out of the biofilm into the entrance of the light organ, which is about the size of a bacterium. So it's like single file into the light organ. And then only a couple bacteria really get in. So it's really just a few, like literally can count them on like one or two hands number of bacteria that are able to get into the light organ, and those are the ones that seed the bacteria that are going to grow up in the light organ.
My understanding, Ruth, and I'm not going to ask you what the volume of a light organ is, but my understanding is pretty much at dawn every day, the squid ejects the majority of bacteria from their light organ, bury themselves in the sand, and the bacteria, which grow very quickly, are able to repopulate during the daytime. Was that your understanding as well?
Yes, that is what happens. About 90%, I think, of the bacteria in the light organ get expelled every single day.
Oh, and by the way, I wanted to tell you, you know how you mentioned that there's this exopolysaccharide involved with this symbiosis?
Mm-hmm.
Shh. That's also true for rhizobium and plants.
Mm-hmm.
So I honestly think that the hand of Darwin has only so many different choices of the ways to do things. And that recognition, that molecular handshake is important. Can Vibrio fischeri colonize other animals besides this particular Hawaiian bobtail squid?
Yeah, so there's actually a bunch of different kinds of bobtail squid. We've got the Mediterranean bobtail squid. There's I think there's one off the coast of Australia. There's also Japanese bobtail squids. They're all over the place, and Vibrio fischeri also colonizes other bobtail squids. There's also some fish that are colonized by Vibrio fischeri, like the Japanese pinecone fish that have the little light pockets by their mouth, I believe, that help attract food, basically, for the fish.
It's really an interesting question. And I say this to you, and I'm hesitant. I know, when am I hesitant to say anything? But I am here. Why bioluminescence evolved? because when most people think about it, they say, oh, because the light that's being produced can be used by a host to do something, say, avoid predators or attract food. And all of that's true, but it's very doubtful that that's how it evolved.
Yeah, we're not totally sure, I think, about exactly how long this symbiosis has been going on. Squid make really bad fossils, so it's hard to tell from like any sort of fossil record, because we can't see how long the squid might have had like these light organs or something like that.
I can add something to that. Bioluminescent bacteria are ubiquitous in the ocean. They can be found in every environment that you care to look at. Additionally, most of those are living within the gut of animals because that's a high nutrient condition. Believe it or not, the ocean is not naturally filled with nutrients. So having these organisms in the gut is fairly common. And then the question becomes, when did that first start taking place? And I would say from the very beginning, just simply because of the nature of where nutrition can be found. But what made light important remains the question. And I know that you and I have talked a little bit about possible biochemical aspects of this. In other words, the light being produced by a bioluminescent organism is just a side product of something else. What do you think about that idea?
No, I totally believe that idea because one can imagine like the very first bioluminescent organism, it probably wasn't trying to produce light. It probably happened on accident and then it turned out to be beneficial, so it was selected for.
Yeah, I think that these are questions that are really hard to answer, and most people aren't interested in them because they would much rather work with charismatic creatures like bobtail squid.
The squid are
It's
pretty
funny.
fun.
It's fun because I knew some people that used to work on the pinecone fish, and it's very difficult to take through a life cycle. And
Yeah.
I know how difficult it was to learn how to do that with bobtail squid as well, but
Hmm.
it was a mess with pinecone fish. So choosing the right system becomes very important, doesn't it?
Yes, definitely.
Where do you think the future of research in the squid-Vibrio symbiosis is going?
Ooh, that's a great question. So there's a lot of things we know for sure about the system from the years of research that have been taking place about I think 35-ish years plus. So there's a lot of wonderful people working on all sorts of different aspects of the system. Um, so there are like people on the squid side working on doing genetic manipulation of the squid so that we can finally like delete some cool squid genes and see what happens with the symbiosis. I think the first goal is going to be to figure out how to get rid of the ink though, because it really messes up our ability to extract nucleic acids.
Just in case people don't know, please explain the ink situation with squid.
Yeah, so squid produce ink in this ink sac that is located actually right next to the light organ, which kind of makes it a pain to dissect out light organs without getting ink everywhere. So the squid produce ink as kind of a defense mechanism. They shoot out ink if they feel scared, if they want to get away. And it's really effective, let me tell you that. I've definitely been duped by some squid ink before, and it does a really good job at hiding the squid. The biggest problem we have with the ink is actually extracting like DNA, RNA. So something like looking at the RNA transcripts of Vibrio fischeri while it's in the light organ has been historically very difficult to do. So I think doing some more higher resolution omics kind of stuff is going to be definitely in the future of the system once we figure out the techniques to do that a little bit better and more high throughput.
Well, you certainly remember rule number one that I brought up in lab is that nature appears to resist investigation.
That is very true.
So I understand that you have some images and some videos that you might want to share. Did you care to introduce those?
Sure. So one of the images I have is of just like a squid in the lab, in the sand. It's got its little like tentacle propped up on the half pipes that we have in there. I thought it was very cute. The squid are just very charismatic. And then I also have this neat little video that hopefully you can pull up of the squid burying itself in the sand. And that is just my favorite squid behavior. It is so adorable to see. It gets everyone. They're like, oh my gosh, it's so cute. And I agree, it is very cute. And then I also have a picture I put in there of the hatchling squid. So these are like day-old hatchlings. They're about the size of a fruit fly. And then the picture I have of them is several of them in a cup. And you can see the little recycle sign for size comparison.
As you can see, this is a wonderful system for people who are interested in developmental biology. It's a wonderful system for people who are interested in microbial genetics, like you and I, Ruth.
Mm-hmm.
And it's been fascinating to see, especially in the lab where you come from, all the wonderful tools that have been developed for working in that system, not just by Mark Mandel, but by other people as well, that have made it a much more tractable system than when people first started looking at it. And it shows the power of people working together on common goals. And it's a wonderful system to be involved in. And I think you've been very fortunate.
Yes, I have definitely been very fortunate. I hope to go back to the system one day because I just miss it so much. And like you said, very charismatic system, glow-in-the-dark bacteria, adorable squid, can't go wrong. And the tools that have been built for the system have just made it so easy to do a lot of different kinds of experiments and ask a whole lot of different questions. So I'm very grateful I was able to experience that for my PhD work.
And of course, they made you a wonderful cake on the day you got your PhD. And there's a wonderful photo of that, if I recall correctly.
Yes, just what the doctor ordered.
How did that feel when you got your PhD?
Oh, it was surreal. It was a crazy month, that's for sure. The month of July was absolutely wild for me. A lot of back and forth because I was also moving over to Minneapolis kind of at the same time I was wrapping up writing my thesis and stuff. So it was just a whirlwind, kind of a blur. But when the day came, I felt ready. I didn't feel particularly nervous about doing the defense. I was just ready to, show people what I've been up to the last few years and celebrate with my friends.
Well, I have certainly been present at some of your talks, and I've read your papers. And I actually posted the one this is not the one that's on bioarchives. This is the one that was published more formally. And can you say a few words about that paper and what you found out?
Yeah, so I published a paper in MBio last year looking at cyclic diguanylate, which is a signaling molecule, and how it impacts the establishment of the Vibrio squid symbiosis. So basically, this molecule, cyclic diguanylate, it modulates biofilm formation and motility in a lot of different bacteria. And if you can remember from my little spiel earlier, motility and biofilm are both required for Vibrio fischeri to colonize the squid efficiently. So what I was looking at was just general trends of what cyclic di-GMP might be doing in Vibrio fischeri during colonization. And what we found is that if cyclic di-GMP levels are really high, it actually inhibits efficient colonization of the squid. And that is a question that we're exploring still a little bit further, trying to figure out what exactly it's doing to make colonization not so great.
Well, and it's interesting to note, we were talking a little bit about jargon, and I want to be very cautious because I think precision is very important, but also it's important to be able to talk about what we do with folks who don't necessarily know the language that we're speaking. Mm-hmm. And science has a language, and well, each subdiscipline does. And you have sometimes to need vocabulary, but that doesn't mean we can't communicate. So what you're studying are the signal molecules that help tell Vibrio fischeri that it's time to colonize, which is certainly very important.
Mhm.
Sometimes some of those molecules have unusual names and all the rest. It's funny to watch people rattle them off at meetings, especially for those 15-minute talks. Would you agree with that?
Oh yeah.
Yeah. Now, I don't think we can show this here because you actually put something or gave a link to something that took me back a few days. When you were taking microbiology from me, there was an extra credit creative project
Mhm.
where you did a beautiful description, coincidentally, by the way, of symbiosis and quorum sensing in Vibrio fischeri and Euprimna. And that's long before you decided you wanted to work with Mark Mandel.
Yes, this is true. Like I said in your micro class, the squid-Vibrio symbiosis is what really got me
Mm.
fascinated with microbiology. So I definitely wanted to do my extra credit project on that. You can see the link in the show notes, I believe. It's basically a little claymation video going over the symbiosis and also going into this thing called quorum sensing, which is how the bacteria talk to each other to make light. So if you're interested, you can check out the video. I've been told by some people at a conference that they use that in their class. So that was pretty cool.
Yeah. And this is something that happens a lot, and I'm not trying to complain or sound odd, but sometimes I receive criticism about some of these creative projects from, some students don't want to do them, some academics think it's silly, and I completely disagree because when you actually do something with your hands, maybe on a screen when you're doing claymation or whatever, you learn it so well. That
Mhm.
creative aspect really, really brings it in. There was another student before you who did a beautiful claymation video of type 6 secretory systems. I'll probably put that one in the show notes as well.
Yeah, I actually got
Um,
the Play-Doh from him. So
well, and I'm going to be leaning on my current micro students saying, see, if you do these projects, you could end up with a PhD of your own.
there you go.
So, Ruth, do you want to spend just a couple of minutes saying what it is you hope to be doing in your postdoctoral work?
Yeah, so right now I am postdoctoral associate in Julia Willett's lab here at University of Minnesota. And what the lab here studies really is primarily Enterococcus faecalis, which is a gut microbe, but it's also a commensal or it's a gut commensal, but it's also an opportunistic pathogen, which means that if it's found in the wrong place, it can cause infections. So, like heart infections and stuff like that. So the lab's really interested in figuring out how it's able to cause these infections and also ways to kind of stop those infections from happening. So what I'm working on right now is looking at an interaction between Enterococcus faecalis and another bacteria called Streptococcus mutans, which is actually the bacteria that causes cavities. So I'm looking at the interaction between those two because they can actually both be found in the mouth. So there could be interactions going on, and there's a preprint from this lab showing that there are some interactions where strep is killing Enterococcus. Um, and what I'm working on is looking at how a different strain of strep is able to kill Enterococcus because it seems like it's using a completely different mechanism.
Oh, that sounds so wonderful for you. And I can't wait to see what the future holds for you. What do you think you would like to do after you complete your postdoctoral stint?
Yeah, so also while I'm here, I'm a scholar with the Aracta program, which is basically this program for postdocs to basically get them more teaching experience and teach them how to teach at the college level. So, what I really want to do is be you when I grow up. I want to be a professor at a primarily undergraduate institution, much like at a place such as University of Puget Sound or a place like that. As I said at the very beginning, small liberal arts schools, primarily undergraduate institutions, are very close to my heart. And I really enjoyed that environment, and I really enjoy working with undergraduate students. That was by far the best part of my PhD, was getting to work with undergraduate students. And I threw in a picture here to show of two of my undergraduate research students at University of Wisconsin, Chandler and Keaton, and they were just really great students. So enthusiastic and so creative. What you're seeing them do in that picture right now is figuring out a way to take pictures of basically my paper lab notebook so that I could put like a digital copy of it somewhere. So they came up with this whole rig system to take pictures easily. And it was just such a great experience working with them. And I want to continue working with undergraduates here at my postdoc and also like for the rest of my life.
Well, I have to say that your students will be very fortunate to work with you, because I've been fortunate to have you be in my classroom and work in my little lab as well. You know, normally, Ruth, I ask people what one event really sold folks on being a microbiologist, but you've answered that a couple of times.
Yes, I have. Mostly you. That's the answer.
Oh, please. In any event, is there anything else you would like to say before we close this out?
Thank you so much for having me on your podcast. I'm so excited to get to be the first Doc Martian on your podcast, and I'm looking forward to seeing what all the other ones might be up to.
I really hope so. It has been such a pleasure to see you. Thank you so much, Ruth.
Yeah, thank you.
This has been Matters Microbial, a weekly podcast about the wonders of microbiology, microbiologists, and microbial centrism. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes for today, including some awesome things, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Ruth Isenberg can be found in the Willett Lab at the Medical School of the University of Minnesota, and I'm so proud of her. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope you've enjoyed being part of our quality quorum today. See you next week on Matters Microbial.